I'm John Pollack, and over the next 10 minutes or so, I wanted to introduce the Caribou community to our newly funded O'Brien Research Center at Stanford. The center's focus is on benign prostatic hyperplasia, or BPH, and its role in lower urinary tract symptoms, or LOTS. The center director is, let me get my pointer. The center director is Jim Brooks, a urologist, and you'll hear him speak at one of the upcoming breakout sessions. And Rob West and myself are pathologists, and we each run one of the main research projects. Okay, so our center builds upon a prior NIDDK P20 grant that we had focused on genomics of BPH. Where we had applied genomics technologies, including bulk RNA-seq, shown here in this heat map on the left, as well as single-cell RNA-seq, to try to uncover new pathogenic processes not known before going on in BPH. And we had a lot of findings, but I think one of the most significant findings was that BPH appeared to entail not just what we think of as an expansion of stromal cells and epithelial cells, but rather what we named cellular re-landscaping, where there was specific expansion of cell subsets and specific depletion of other cell subsets. So building from those findings, one of the overarching goals of our center is to create a comprehensive BPH atlas, which would span multiple scales. So from tissues down to cells, down to individual signaling molecules. And to achieve that, the center is structured with two main research projects, which I'll discuss briefly, as well as two cores, an administrative core that oversees the center and educational and training opportunities, as well as the Biospecimen and Bioimaging core. And so just a slide on that. So the purpose of the Biospecimen Bioimaging core is to procure needed prostate specimens for investigators, to provide tissue characterization services, and to archive MRI and digital histology or immunohistochemistry images. And I'll just the next slide, I'll just talk briefly about a couple of those characterization services that the core provides. So these are services sort of illustrated here to support the research going on at the center. And both of these technologies were pioneered by groups at Stanford. In fact, pioneered by our groups at Stanford. So on the left, what's shown here is spatial transcriptomics by LCM Smart3Seq. And this was a technique developed by Rob West in his lab. It's a technique for spatial transcriptomics where minute cell numbers are laser microdissected, captured, and then that low input RNA is sequenced with deep Illumina sequencing. And it can provide a full transcriptomes on very small numbers of cells, actually down to individual cells, as shown here in this, I think, a PCA analysis. Sorry, it's a TS&E analysis, but so on the right, what's shown here on the right is another technology also developed by our groups, in this case by Michelangelo, no relation to the artist. It's a platform for multiplexed immunohistochemistry, and it can simultaneously quantify as many as 40 or more different targets in the same tissue section. And it does that by labeling the antibodies not with fluorescent dyes, but with heavy metal tags. And so those images and those antibodies are then resolved by mass spectrometry. Okay, so leveraging those two technologies and others, Research Project 1 focuses on the role of stromal fibroblasts in BPH pathogenesis. So in work that we had previously published, we found that signatures of stromal fibroblasts correlated with patient symptoms BPH symptom scores, as shown here on the left. So that underscores the clinical relevance and significance of the stromal fibroblasts. We also found by techniques including single-cell RNA sequencing that BPH tissue was enriched for specific fibroblast subsets, including what's shown here is a subset that overexpresses bone morphogenetic protein 5 or BMP5. So there's enrichment of specific fibroblast subsets, but at the same time, we also found evidence of depletion of other fibroblast subsets, including a fibroblast cells that sit near the epithelium that are positive for expression of estrogen receptor alpha that we see in normal prostate epithelium, but are mostly absent from the stroma underlying the BPH epithelium. So building from those data, the broad aims of Project One are to define the fibroblast subsets in BPH in comparison to normal prostate, to determine the key interactions between those BPH fibroblasts and the overlying prostate epithelium that drives prostate enlargement, and to distinguish between BPH stromal fibroblast origins in embryonic reawakening, a decades-old hypothesis, versus injury response or inflammatory response. Okay, so Project One is the fibroblast. Project Two focuses on the immune microenvironment and its role in BPH pathogenesis. Shown here on the top left from bulk transcriptional profiling, we had found that the single largest transcriptional induction was of CXCL13, which is a chemokine, which is also a lymphocyte attractant. And single cell analysis shows that CXCL13 was produced by BPH fibroblasts, that same fibroblast subset that was also enriched in BPH tissue. What's shown here is that CXCL13 levels indeed do track well and correlate with B cell or T cell signatures in the BPH tissue. So building from those data, the broad aims of Project Two are to define the immune cell populations in BPH by the multiplex immunohistochemistry, the MIB-TOF, the mass spec method, to understand the function of those immune cells in BPH or driving BPH. And lastly, to determine whether that CXCL13 overexpression and the accompanying immune cells are part of an immune response or else a senescence response. Okay, so that's a quick tour of our center and what we'll be doing over the next few years. Since I'm speaking in the collaboration session, I wanted to end by discussing potential areas of future interactions and collaborations. So we'd very much like to collaborate with other centers or individuals outside of within the sort of broader benign urology research community that share our interest in BPH and LUTS. Towards that end, we've already begun some discussions with Doug Strand and Will Rickey of the Wisconsin O'Brien Center. So please reach out to us if you feel you have shared interests in some of the things that we'll be doing. Those collaborations might well entail utilizations of resources that are unique to our center, including some of the tissue microarrays that we're building, the spatial transcriptomics by laser microdissection coupled with Smart 3-seq, and the multiplex IHC platform based on mass spec, the MIB-TOF. And there's also collaborative opportunities for trainees at all levels. So with that, I'll end and thank you.